All right, I think we're going to go ahead and get started. So hello, and welcome to Structuring Biotech Data for Machine Learning and Artificial Intelligence Success. Thanks so much for joining. My name is Bernie Lee. I'm a product manager here at LabKey Software for our biologics LIMS. So at LabKey, our experience and expertise comes from helping academic, government, pharma, biotech, and a variety of other institutions manage scientific data for over 20 years. We aim for genuine partnerships with the groups that we engage with to ensure that you achieve your goals and that we can learn from you in the process. And as you might have guessed, many of our partners build or have built sophisticated data science products that interact with data in our systems. One of these products that this is done with frequently, especially in the last few years, is our biologics LIMS. Which I'm gonna mention a little bit more about near the end of the presentation. So the primary objective today is to help you structure your data for increased efficiency and accessibility now, and future-proof it for better utility later. We typically find that groups have very labor-intensive data operations, analyses, project decision-making, and retroactive inspection of data. And making use of data beyond the immediate analysis can be really, really slow or limited because of its poor organization, integration, and structure. If you don't address these issues early, you will really wish you did when you're ready for data science applications. So let's start with why all of this matters. Many of these data science endeavors, AI and machine learning applications, are really cool. And for some, maybe that's enough of a reason to do them. But they also offer some serious potential value. While it would be nice to be able to just say something like, I want an antibody that modulates my system of interest, some particular target, and you'd get back the sequence. You could just go synthesize and confirm that it does what you want. What many of these machine learning tools actually do is de-risk the lengthy and expensive R&D required for success, predicting and heading off liabilities in preclinical and clinical work, and ultimately reducing failure rates. These sophisticated endeavors that you all are on are expensive and time-consuming, and machine learning applications can help generate candidates or predict what might work. Alternatively, they can at least eliminate what will not work, thereby reducing the cost and time of development. And my last selected benefit here is about learning primarily and operations. When you structure your data for AI, for machine learning, and other data science applications, you will undoubtedly gain insights that would be otherwise obscured by the lack of organization in that information. So before we jump to some of the key challenges and best practices for pursuing this kind of project, have a look at the handful of examples that I've selected here from around the industry, a very high level, of course. I selected these to share just in the hopes that they provide a little context. But if you go looking at various publications, every month you'll find additional ones. So there's an incredible number and range of these applications. But what I want to focus on here is what all of these data science products share that make them successful. What does it mean to be successful in an endeavor involving machine learning, AI, other data science applications, large-scale analyses that might be applied in biotech? It's actually simpler than you might think. We define success as scientists can spend most of their time using their specialized domain knowledge and very little on moving and transforming data around or other data-related menial tasks. Success means having clear metrics demonstrating time savings or added superpowers from some modeling product. There's a role for data science here in driving down the cost of many data science endeavors by reducing or replacing experiments and reducing clock time. But it is critical in most cases to keep the true goal in mind and not, as I and many others are so inclined, to pursue the really cool project for its own sake. So why is this hard? I know it seems like everyone is doing this, so it must be easy, but it is not. And even if you've succeeded to some degree with one project or another, there are probably ways to make it easier the next time. Architecturally, unlike more common rules-based programming software development, machine learning consists of two primary parts that make up a final executable algorithm. There's the machine learning algorithm itself, and the training data to learn from. Your files, your raw files, are typically not ready to be used directly in machine learning models. So data preparation is very much at the heart of this. And it's this preparation as well as scientists' processes that's worthy of our focus. You'll hear this all over the place, but it's true. The preparation might be more important than the algorithm itself. That's certainly true of time spent. In part, this is because there are a lot of added benefits that come from that preparation. So for data to be useful beyond a scientist's immediate needs, regardless of whether it's machine learning as the ultimate destination, it really needs to be structured, consistent, and interoperable. So structured data is anything that conforms to a set of rules and conventions, with a good example being data that can be naturally cast into a tabular form. Unstructured doesn't mean poorly structured, like having the same info in tables with different headers and naming conventions. Unstructured can be text without defined patterns, mixed information, or images which are inherently unstructured. You can process all your data on its way to the model, but data science models are very performance and resource sensitive. The more processing one must do to train and run the model, the more challenging it's going to be to find that success. I don't have to tell you what consistency means, but if everyone is doing things differently, or if the same people introduce variation into their data, making use of it after the fact can be extremely difficult. And interoperability is the ability of software to exchange and make use of information. I typically think of APIs or direct database access, but of course, not everything will be in a relational database. I assume most of you are or were scientists. I was for many years. Scientists will always find ways to accomplish what they need to with their data. It's, scientists, we have to have high ingenuity to be successful almost definitionally. But in this context, it often means falling back on the tools that we know, usually spreadsheet operations, when alternatives to it are difficult. And this can introduce poor structuring. It implies that whatever data management tools are employed to support these efforts must therefore improve cycle time for scientists, or at least not slow them down while providing benefit elsewhere. So different teams, projects, and people will structure differently what is essentially the same data. For example, groups may use different templates for the same thing. If they don't have cause to communicate and get aligned, this will result in different formats between projects, which introduces inconsistencies. And then where does the data end up and in what form? Is it the same between teams? Does it end up somewhere interoperable? An example, do scientists drop result files on their own computer or in their own shared folder space? Is the data imported into a database somewhere and in a form that's accessible, recognizable, and therefore useful to others? If the answer to any of those questions isn't what you might want, this will introduce interoperability issues. Volume. Let's talk about data quantity. You want the amount of data necessary to solve the problem at hand. Is more better? If you need to detect small, meaningful differences, you'll certainly want to pursue the difficult aim of getting a sufficient quantity of quality data up to some point of diminishing returns. But those diminishing returns are very real. For example, we know that everyone's favorite AI chatbot was trained using text databases from the internet, thought to have been around 300 billion words, 570 gigabytes-ish. Would a lot more have been better? I'm sure they debated. There would have been some advantages and some disadvantages, but it accomplished what they wanted to accomplish with that first round. On the other hand, if you're preparing data with particular tasks in mind, having a huge amount of imperfect data can demand a huge amount of work. It's often better to start small and reduce the complexity of your data, and perhaps more importantly, tailor your data gathering mechanisms toward the desired ends. All that said, you definitely want to collect all data that's possible, but biotech doesn't often have stereotypical big data problems. We most often have discrete runs of data generated by rate limiting instruments. The way we increase data quantities is with things like high throughput technologies. Then there's diversity. Do your operations truly collect 1,000 different types of data? Probably not. Most often, real variation in data types are artificially expanded by different people or groups interacting in different ways with similar data. Sort of like the examples that I shared before, meaning that it sort of masquerades as diversity. You might even call it inconsistency. So as an example here, you might have multiple groups running extremely similar assays. And they may name it something totally different, keep it in separate places, and name some of the attributes differently. But in reality, there's very little or no difference between the two. There's a column or two. And failure to get a process in place to check that similarity and adjust with future use in mind leads to an artificial inflation of data diversity and complexity. This in turn leads to hurdles for large-scale analysis. And then there's complexity. Some types of data do not lend themselves well to structuring. They require heavy processing, transformation, or conversion to be structured and integrated. These are things like images, mass spectra and chromatography, transactional data like sensors for continuous monitoring of bioreactors. You can transform these into something more structured that supports the end goal, but there are trade-offs. For example, a modest pixel count JPEG that's well encoded is going to be about 13-ish kilobytes of storage, whereas a float 32 tensor, which is great for machine learning, of the same image data will utilize 700, 800 kilobytes of memory. So like a 6,000% increase. It's a pretty significant trade-off, especially when you're doing these things at scale. Okay, so let's say you've overcome most of these challenges. In order for some model to be useful, many of your data types need to be integrated to produce a contextual landscape that can answer scientific questions. Very often, scientists are manually combining results with sample data, experiment conditions, upstream data about sequences, clones, and other sources like that, and then using the results to make decisions and keep records. To use a more concrete example, if you're tracking machinery sensor readings on a bioreactor to enable predictive maintenance of your systems, most likely you're generating logs of transactional data. So that's like events which snapshot specific moments in time. But you can add in the equipment model, batch information, its location, in order to look for dependencies between equipment behavior and its attributes. You'll also, when you use them, draw samples with information about them, which needs to be associated with assays characterizing them. There are separate systems to collect high-frequency sensor data, things like pH, gases, scale weights, which are very relevant context for the samples and the ultimate experiment success. And then you have to take all of these pieces and integrate them to make good determinations on broader topics like cell culture health and bioreactor operation. And this means, among other things, you need to align identifiers and timestamps, and then store it in such a way to support programmatic accessibility and usability. All of this is no small feat. It's a lot of different types of data in different forms and shapes that you need together in order to truly make a lot of these determinations. So the last key challenge I'm going to mention is institutional and structural support, which possibly precludes overcoming all the other challenges that I've mentioned. at least on an institutional level. So institutional authority, support, and structure are required to organize, coordinate, and implement changes needed to successfully use data science products. Many of these aforementioned challenges, part of higher order data work, are simply beyond a reasonable scope of responsibilities for many scientists, IT professionals, and software developers, especially since they require changes which will impact everyone's responsibilities, processes, and daily operations. So organizing your institution to support these efforts can be a precedent required for success. And as part of that, data science, data engineering groups with data management tools are really valuable to have at a biotech company or endeavor. Okay, so we've reviewed some of the key challenges. Now it's time to start solving the problems. This begins with a clear problem formulation, which will provide guidance on what data to capture and how to process it, and what additional work will be needed for the model. It'll begin with the people that a data science product might serve. So as often as not, your scientists will come to you with these problems, but if they don't, begin with the people that have the domain expertise. It is critical to incorporate domain knowledge into the data structuring, processing, and conclusions. It's their knowledge that will help you select relevant data features, understand and neutralize potential biases in data collection, and ultimately help judge utility. Let me tell you a quick, well-known story to make the point. So there was this healthcare project aimed at cutting costs in the treatment of patients with pneumonia. It employed machine learning to automatically sort through patient records and decide who has the lowest death risk and should just go home and take antibiotics, and who is at a high risk of death from pneumonia and should be in the hospital. The team used historic data from a variety of clinics, and the algorithm was accurate. But there was an important exception. One of the most dangerous conditions that may accompany pneumonia is asthma. And doctors always send asthmatics to the intensive care unit, resulting in minimal death rates. So there was an absence of asthmatic death cases in the data, which made the algorithm assume that asthma isn't as dangerous during pneumonia. And in all cases, the machine recommended sending asthmatics home, despite having such high risk of pneumonia complications. So not all data scientists know that asthma can cause pneumonia complications, but a quick conversation with the domain experts, a doctor, would. If you and the model have incomplete information, despite terabytes of relevant data, a machine is going to be nearly useless, or perhaps even harmful. There's now a commonly used simile that being a great data scientist is like being a great chef. To create the desired meal, you must possess great familiarity with all the potential ingredients, in our case, all of the potential data, so that you can understand how to prepare and combine them in a complementary fashion to produce a balanced and memorable dish, or in this case, a high-priced machine learning tool. So with clarity around the problem you're trying to solve, you can begin to lay the groundwork for that meal or for that successful machine learning model. And you want to consider the whole data lifecycle in order to do this. How is it typically captured, processed, analyzed, and stored? If you can clearly identify what will happen at each of these stages, then you can select the right tools and places for these operations to happen, so people don't fall back on extemporaneous and variable spreadsheet use. I want to briefly emphasize the analysis part of this data lifecycle, because really this is how the fundamental scientific questions are answered and where most of the data manipulation occurs to achieve it. So it's towards these aims the data is transformed from its original state, annotated, integrated, calculations are performed, visualizations are generated, and decisions are often made. And it's these questions that often lead to good data science projects. Okay, we're going to discuss some nuts and bolts here. This is going to be a high value sampling and high level sampling for lack of time. So let's start with structuring and standardizing your data. There are a variety of practices that can help you stay sane as well as make your data science endeavors much easier. They center around choices that your organization can easily make with little forethought, with a little forethought and coordination. So as some of my previous examples have suggested, adopting a common data model shared across projects and across teams can be the foundation for consistent structuring. So within that framework, ensure that your data types are appropriate and consistent. So a silly example is the money variations that I have shown in my second bullet here. These are all legitimate ways to write $4.03. But some of them are numeric, some of them are strings, some of them have special characters. Make a choice together about what form these are going to take, and then stick with it. I will say that most data management tools will store it one way, but allow you to call it a variety of things, to alias that field. This is a case in point, though. The data management software is enforcing a standard within itself, even if it accommodates your variability on input. So you want unique IDs for entities of interest. This is fundamentally how data scientists and frankly, you and your scientists make distinctions. If you cannot distinguish between unique entities, there is no analysis that can be done beyond the immediate data, and certainly nothing a machine learning model can do for you. You're also going to want to avoid totally free text. There are 170,000 English words in use that produce an effectively infinite combination of free text entry possibilities. That's not good. Find some way to constrain the choices for information entry. Even if the list is long, having standard known choices makes the data usable. Then there's validation rules. Validation rules ensure things like ranges of data are appropriate. So bad values are kept out, outliers are excluded or rectified. A few quick examples will paint the picture. What is an appropriate age range for a human? The oldest human ever died at 122.5 years. So hopefully we can agree at something like 0 to 130 years. That means you should not accept ages that are 4 digits in length or negative. Similarly, there are only 12 months in the year. I'm not aware of temperatures below absolute zero. Food is either raw or cooked. I think you get the picture. But there are also custom validation rules, things like dependencies on other data that dictate what is valid here. Keeping data clean and consistent through validation is one of the strongest ways to maintain data quality. Okay, as I said before, integration of your data creates the landscape from which scientific and practical questions can be answered. And alignment on data types is really important. So imagine you have 2 types of data for temperature. One is numeric with one name, and the other one is text with another name. Now, any data science model that cares about temperature is going to have to somehow reconcile those on the fly, which is not good. What you want to do is convert to the same type after deciding whether you're going to call it T, temp, temperature, degrees C, something like that. Next is key alignment. Typically, multiple rows from datasets all relate to the same entity or observation. One or more key fields is used to tag all data associated with that entity. And a common challenge is key alignment, where the key values used to tag the associated data don't match across all of those data sets. So for example, different data types or formats might be used. You can have things with leading zeros or trailing spaces, variable capitalization, punctuation. And if you fail to fix these issues, it can actually lead to significant issues, inaccurate analyses due to the introduction of fictitious observations or the loss of data associated with the true observations. On a case-by-case basis for local analysis, this can usually be repaired, but it can be quite catastrophic for machine learning, which is typically done much later. And lastly, you will have missing data. At best, when you start integrating and combining data from many experiments, you'll find that some assays weren't run, and probably for good reason, that you and your scientists know. Some sample information might not have been captured, again, possibly for good reason, and that these and similar gaps are much more glaring for data science than they were for the original experiments. And interestingly, the best practice for machine learning specifically here is to fill in those values for algorithm training that are appropriate. It turns out to be a lot better than no data. Okay, let's talk about the importance of metadata. The other dimensions of data I'm referring to as metadata. They provide the context and conditions for key measures. So metadata is important because you don't just want to know the optical density of some sample, which might be the key measure. What you want to know is how those samples compare to each other and whether your conditions allow you to make that comparison appropriately. So you want to know how something upstream of your measure affects your materials. These simple questions require adequate contextual data. As you move into data science products like machine learning, AI, and other large-scale analyses, the richness and accuracy of this metadata becomes paramount since it shapes the model itself. To use an example, you have a bunch of amino acid sequences and calculated physical properties that are associated with them. And you want to be able to predict what purification method or methods you ought to use for future sequences. So by testing under various conditions, the various resins and pHs, you can learn what yield and purity you're going to get for those molecules. Now, if you can model it right, you can extrapolate from similarities between the training sequences and other properties what a new sequence most likely needs. Now, in that case, you obviously need the annotated sequences, the pH and resin information, but you also likely need to capture starting quantities so you know what your yield means. You need probably information about the cell line producing these molecules, and various reagent information that's used in the process. If you fail to capture that metadata and something important varies, it can categorically change the downstream measures, and the model will simply be incorrect. No matter how much data and expertise you have, it will not compensate for incomplete context. Again, I'll remind you of that pneumonia asthma example. And then briefly here, what form do you want your data in for the purpose of data science products? There's actually not a universal format that magically facilitates machine learning. It really depends on the problem you want to solve, the kinds of data you have. And this inevitably leads to transformation requirements to get the data in forms optimized for machine learning. Because it's not universal, what I can help you think about is finding the data, regardless of how it's stored, whether it's pointers to files and metadata or some relational store. It does need to be stored in such a way that there's a query mechanism. You have to be able to ask the system for certain data in a certain form. And if you can't do that, then you'll eventually just have to do all of the manual data gathering, and it's quite a nightmare, especially at scale. I also want to give a quick plug here for Python, the programming language. The Python language has a lot of built-in mechanisms to help with many operations and transformations necessary for machine learning. And that means that if you can use some data management tools that offer a Python query API, for example, you should. And of course, no talk like this would be complete without at least one mention of automation. I'm not talking about robots to improve your throughput here. I'm talking about automating components of the data lifecycle to collect more data that is cleaner and more reliable, because this can be the expensive part. The cleanest data is the stuff unchanged by a human. I know that's a big surprise. If you can start your automation process with the raw data, do it, be happy. If you're reliant on people to enter information, you will be rate limited and may have accuracy and consistency issues. For example, when a scientist has a spreadsheet in place and you have to identify what's in there, what's a calculation, what's copy and pasted, what's sourced from somewhere else, it can be very difficult. The automation allows you to process and analyze data consistently, and ultimately store it exactly where you and everyone else expects it. So this will make everyone more efficient and far less frustrated when older data needs to be found and used for something other than its originally intended purpose. So consider what you can automate, consider what impact that will have on your scientists, and consider what impact it may have on your institution as a whole. And that brings me back to institutional support. So scientists will understandably find the lowest energy approach to their own data management needs. And imposed data collection, processing, and storage tasks may be tedious and burdensome, even overwhelm them when added to all of their other responsibilities. So if people must constantly manually make records, the chances are they'll consider these tasks as yet another bureaucratic whim and let the job slide. So to return to our success criteria, have a team whose goal it is to ensure scientists can spend most of their time using their specialized domain knowledge and not pushing data around for themselves or others. That might mean hiring data architects and data engineers. Deliver value to scientific staff effectively, in part by developing clear metrics demonstrating time savings from some data science products that are enabled by your efforts. Use data management tools intended to govern the data lifecycle. You do not have to do this alone. You do not have to write all your own tools. And work with leadership at your institution to create a data-driven culture and prevent data fragmentation. With a better understanding of these fundamentals and best practices, you may grasp that this will be hard without the right tools. Our answer is LabKey's Biologics LIMS, which accelerates discovery and development processes while automatically structuring and future-proofing your data. It's a cloud-based lab information management system tailored for emerging and scaling biotech companies. The system links samples, plates, assays, biological entities, analyses, and experiment documentation in one central location without heavy spreadsheet work. Here are some of the feature areas for large molecule R&D. We would really love to show it to you and learn from you. You'll find a link in the chat and also in the follow-up email after the webinar. I want to thank you truly for attending today. And if you have any questions, I'd appreciate it if you would submit them and then stick around for a few more minutes. Thank you all so much.